Good afternoon, ladies and gentlemen, and welcome to the new season of challenges in forest ecology. We have a great program again this year, or certainly for the first half of the year already organized. It's my pleasure today, my real pleasure, to introduce one of my long friends and colleagues, Boris Rewald. Boris is an associate professor of ecophysiology of plants. He works at Mendel University. Boris first began in research not that long ago, 2000 and he worked at University of Göttingen, where he got a PhD. And about this time, he started to work on roots and competition between roots, which was at its time groundbreaking. Nobody else was doing this at the time. He then went on to spend a considerable amount of time, about 4 years in Israel, working on dry land research, again on roots. And then he moved to Brno, where he spent the best part of 10 years, and now at Mendel University. As I say every time with all our speakers are highly cited. Boris, for him, still a relatively young man, although he's going a bit grey, but still a relatively young man, has an H index of 40. He's been cited over 7,500 times, including one paper which has been cited 1,500 times in about the last 3 years. So a quite remarkable career so far. So Boris is going to talk about mixed tree plantations, the good, the plastic, and the uncertain. So Boris, your screen.
Thank you very much, Douglas, and I'm glad that you made it possible that I also present here in this esteemed audience. So welcome everybody, wherever you are. Good morning, good afternoon, or good evening, because particularly our dear friends from the Asian Root Project even joined from Thailand, and it's pretty late over there. Let me share my screen a second. So I like to circle back basically to my beginnings in Göttingen about root competition. It's not implicit in the title, but I thought it's attracting a larger crowd, because sometimes the roots can be a bit scary, because it's still the hidden half, and we are not everybody is so comfortable to deal with roots. So I like to talk about the good, of course, because we talk about mixed forest, and nowadays, to rephrase Bill Clinton, it's all about mixed forest, stupid, yeah? Because we expect great things from mixing, diversifying our forests to make them ready for future climates. And among the very good things I want to introduce first, maybe also to the persons which are not so familiar with the topic, is about great things we expect from this mixed forest in regard to biodiversity conservation, soil carbon sequestration, for example, or generally maintaining high productivity under unstable climates. And of course, there is increasing evidence there are many people now interested in mixed forests, and a lot of studies going on, including the tree DiffNet studies. I was happy to be involved in with the bee tree. And we see, for example, the mixing effects on here, for example, understory plants diversity are generally positive. Yeah, this was a very nice review last year. But of course, if we have a closer look, and also very nice review from the Jürgen Bauhaus group in Freiburg, for example, we see that these diversity effects, of course, don't materialize in all kind of mixtures. Yeah, it's very impressive to see, for example, here this understory plant diversity. Only in 40% of the cases, for example, there is a positive effect. Yeah, just to keep this in mind, it's not a universal solution. If you look below ground a little bit, and there's in general, of course, less data available, we see that they're even less positively affecting soil mesofauna or soil microbiota but still in a considerable amount. And we could last year confirm this. So Ramona Werner and Joel Jensen were the lead authors on this tree-definite publication, for example, where we looked not only, but also at the effects of 9 sites on the tree diversity or effects on the mycorrhizal or fungal richness, for example. But what you see here is that, of course, these fungal communities, they cluster strongly along environmental gradients. So pH, CN, and mean annual precipitation or something like this. But in general, for example, in this experiment, we didn't find an overall effect on the fungal diversity. And you see even on the right-hand side that the Shannon index of the ectomycorrhizal diversity declined, which in the highly diverse forest patches, which means about 4 to 6 species, which means they're getting more similar under increased diversity. Nevertheless, this mixing effect had positive effects on other ecosystem functions, not only habitat. For example, here the carbon storage. You see that about the highly diverse forest here in this gradient from France to Scandinavia had about a 10% higher topsoil carbon stocks. Yeah, these are all European forests. So this is a significant amount, also considering that these plantations are relatively young. Similar interesting studies are, of course, going on worldwide. This is a nice paper from Florian Schnabel, also from Freiburg, where they nicely related tree architectural changes, canopy opening, the effects on this on the herbaceous vegetation and carbon litter on soil organic carbon stocks. So there are all kinds of studies going on worldwide, trying to explain basically what are the positive effects of mixed forest and why are they materialize. Last, not to forget, of course, there is the large productivity effects of mixed forest. For example, this is also a result from Steinparzer et al. From the bee-tree experiment, where we see that, for example, there is a clear over-yielding effect on the leaf area index. On the left-hand side, you see single species mix, single species monocultures, Acer, Tilia, Quercus, and Capinus, 2 species mixtures, and the 4 species mixtures. And in the center, you see already with the star, the observed, with abbreviated with the O, and the predicted values. So in most cases, this leaf area, specific leaf area index is higher than expected from the monoculture. So obviously, there is very nice complementarity of different species, and then an increased productivity of the sites can be achieved. And the same holds true for stems. Many studies now show that there is a higher productivity in mixed stems, and also in 2024 was in a large community effort. Cheng et al. Showed that the community productivity here as the basal increment per hectare significantly increased with the species richness, and this changed even positively with the ongoing age of the forest stands. Of course, we try to explain these changes, and these changes very common now, and I'm also a big fan, is a trait-based approach where we see there are different properties of plant species, and we relate this growth or other ecosystem functions to specific traits. And for this growth analysis from Cheng et al., for example, we showed that the shift in the contributing species together with the shift in specific traits, for example, above ground traits, specific leaf area or the leaf nitrogen content decreased over time, while for other species, with a lower specific leaf area or lower nitrogen content, it increased. So there is a shift in the community level, basically, in which traits contribute much, and this could explain basically this change or increasing temporal resolution of the overyield. Now, Douglas introduced me nicely as a root researcher aficionado, and this is indeed because I'm a bit worried about that we see a lot of effort to relate these mixing effects related to above ground traits, and we really made great progress in relating different ecosystem functions to specific above ground traits, also relating litter fall or microbial communities to soil organic carbon. But what's very often missing because of the labor intensity of the work is the fine roots, because really fine roots is a missing link for me. And if you see on the right-hand side, why is it so important to study the traits and the frequency of certain traits in a community? Because this basically underlies a lot of basic ecological functions here, for example, illustrated with the stability of ecosystems, where we could have the community weight means of a certain species contributing, for example, to the stability of a stand. We have the functional diversity, also of course coined by the trait profile, or we have a redundancy of different traits in my community, underlying an insurance hypothesis. So I want to separate this talk actually in two parts. So we have a biomass allocation part, where first I want to talk about which effects mixing has on root biomass and its dynamics. And in the second part, I want to have a closer look at different root traits and their plasticity. So which traits are actually plastic and to which degree from our findings, and how does this different or this change competition, mono intraspecific competition between species of the same species in monocultures, and competition between intraspecific species, so not different species in mixtures. And to illustrate this, I use two different experiments. One experiment is the bee tree experiment in Tulln, one of the 3 different experiments. It's a relatively young forest stand, so planted in 2013 in eastern Austria, and there were 4 different species intermixed. So all of them deciduous. We had monocultures of Acer, Tilia, Quercus, and Capinus, 2 species mixtures. Because of the space restraints, only Acer and Tilia, and Carpinus and Quercus. And we had, of course, 4 species mixtures where all 4 species were equally distributed. So we had 100% monocultures of one species. In the 2 species mixture, we had a 50-50 distribution, and 25% of each species contributed in the 4 species mixture. And as you might know, that these species differ in their ecological strategy. So we have the very acquisitive, fast-growing species like Acer in there, and the relatively slow-growing and conservative species like Quercus. And this will maybe matter a little bit how we interpret the results. And the second experiment I did about in the same time. It's a root competition experiment in mesocosms. Maybe requires a little bit more detail to explain. There we have large pots, which we see here, labeled here as plant 1 and plant 2. And they were planted in unfertilized soil. And these large pots were connected with 2 competition chambers. One of these competition chambers basically was fertilized, here labeled as plus NPK, and the other one was also fertilized but without nitrogen, so only P and K. So the beauty of this is basically we could manipulate the different competing competitive environments by inserting from either side basically competing roots. And we did so either by having only one root inside, which I call ISO, so it's an isolation treatment, no competition. We have intraspecific competition, means from both sides, both plants were the same species and 5 centimeter were entered, and we had interspecific competition where different species were entered into this competition chambers. And the beauty here is, of course, because we have so much control, we can relate directly the growth of one root with a different one. So is this relevant that we look at the effects of root interacting actually in normal soils? Yes, it is. So on the left-hand side, you see the biomass distribution in common course in this bee tree experiment, and you see that in most locations, or in all locations, basically there is a large overlap in the root system. So there is a root system interaction. So they're not segregated. Root system segregation you very often find when you mix gymnosperms and deciduous ones. So is this also realistic in the field? And this is actually, it's nice to show, this is my first publication in 2009. Rewald and Leuschner from a dry forest stand in central Germany, also 4 species. And there is the frequency of cores with 1, 2, 3, 4 different species in there. And you see also in the mineral soil, most of the soil cores had at least 2 or 3 different species inside. It's very rare that you have only 1 species. So mixing is rather the rule than the exception in this deciduous mixed forest. Now the first results, and you will see that the data from the Turin experiment from bee tree has always a blue strip on the side, and the data from the microcosm experiment has always a gray outline, so you can easier distinguish them. So look at the biomass effects of this mixing. First, the conventional one, what we know already from many experiments. So there is, of course, a significant over-yielding in the leaves. Yeah, this is leaf biomass. It's always indicated here with stars. Yeah, so APTC, so the mixture of Acer and Tilia, for example, produced significantly more leaves than expected from the monocultures. And the same was true for the 2-species mixture of Quercus and Capinus, and also the 4-species mixtures. Similar for the wood biomass in the center, you see APTC, and the 4-species mixture had a significantly higher stem or branch biomass. So of course, this talk is about roots, so we were very happy to see also for roots, there is a significant over-yielding below ground, so there is more fine root biomass in the 4-species mixture observed than expected. This could be, of course, important for any kind of carbon input into the carbon sequestration. How much litter could be theoretically transferred to the soil. And if you look more closely, you see that this distribution of biomass below ground is not necessarily following the biomass distribution above ground, and it's also not particularly related to the frequency. Yeah, again, monocultures 100% of one species, 50-50 mixture in the 2 species, and 25% of each species in the 4-species mixture. So we were very, very happy, and we thought we explained the different frequencies of one species by its competitive ability. Yeah, if you look closer, for example, by QRCB, you see that the yellow bar, this is the Capinus, yeah, the hornbeam, has significantly higher biomass than, for example, oak roots. Yeah, and similar in the mixture of Acer and Tilia, you see that Acer is dominating the fine root systems, and so the mean trait values in the Acer and Tilia mixture. Yeah, and this we of course thought is somehow related to their competitive ability. So there is an active, foraging approach to claim the soil for resource uptake. And then we were a bit puzzled, and this is also one of the reasons why this is still ongoing publication with Ramona Werner, because if you look at the left or the right figure, you see there's a relationship between the above ground biomass and the below ground biomass. And on the left side, you see basically that for Acer, only for Acer, there is a clear significant linear correlation between the above ground biomass in the below ground biomass. Of course, across all monocultures, as expected, larger trees have more roots. It's true, significantly true, but also for Acer. So Acer obviously doesn't have what we often call ontological scaling, which you see on the right-hand side, because we know from previous publications that many trees have a lower root mass fraction as they get larger or older. And so basically you see for the other trees, the more conservative species, Quercus, Tilia, and Capinus in the mixture, that the total biomass scales 100% with the root mass fraction. This allows, of course, the question if actually there is a limited plasticity in this biomass allocation under competition. So is this scaling we saw before, this variability, this root placement, really an outcome of interactions between the root systems, or is it merely reflecting basically the different sizes of the trees in the community? Yeah, so it's always a question for me, what is an active acclimation process and what is a passive response to general growth pattern? If we look now in the mesocosm experiment, so there we see, first of all, on the top, always labeled A, and the bottom labeled F, there are also not very significant effects of direct competition on the root biomass. Yeah, there's always labeled here ISO. You see, of course, the variability of the root biomass in Acer, with very large amounts partially, is very high in isolation, because obviously this root had the full 1 liter of space of the competition chamber. But in general, the means did not change significantly according to the root biomasses. And there I want to acknowledge, of course, something we often forgot when we sample in the field, like in bee tree, that we cannot make this distinction between isolation and intraspecific competition, because even that we sample in monocultures, we rarely check, or I don't know any study actually, which see how overlapping are the root systems of neighboring species, yeah, or neighboring trees of the same species, yeah, just because we don't apply the molecular markers maybe to check this. Yeah, so this, what we call monocultures in field experiments, is basically always, in my opinion, a combination of isolation and intraspecific competition, which makes it, of course, hard to distinguish the effects, potential effects on traits. If we look or continue a little to look at the fine root biomass, we see here on the left-hand side the distribution curves for the fine root biomass under isolation, intraspecific and interspecific competition. For on the left-hand side Acer, yeah, again labeled A, on the right-hand side Fagus. And what you see actually for Acer, you don't see much. There's not much plasticity, neither in terms of variability nor in the mean, either under intraspecific or interspecific competition. On the right-hand side, you see that for Fagus, yeah, for Fagus, I don't know if you see my pointer actually on the screen, you see that there is indeed a change in the variability. Yeah, so the fine roots of Fagus become less variable if they're under interspecific competition. Yeah? Under high nutrients, NPK, this is true for both intraspecific and interspecific competition. So in itself, they become more similar. So there is less variability in the biomass distribution when they are under interspecific competition. But on the right-hand side, we see the effects of the different nutrition regimes, where you clearly see in green the NPK-grown roots and in purple the PK, yeah, so without nitrogen fertilization grown roots, where we see the determining factor here seems to be the nutrient availability in a certain hotspot. And this is, of course, relevant because we know that soil is, yeah, because we work in forest soil, it's not agriculture soil, so it's relatively heterogeneous. Yeah, but for me, it's unknown basically the degree of heterogeneousness in mixed forest, and yeah, we come back to this later. Now, this experiment gives a great opportunity to compare pairs, yeah, because it's a pair design. In this figure, you see the direct effects of plant 1, or target plant, the competitor plant, on the target plant, plant number 1. So basically, two opposite plants in the mesocosm system, which had their roots connected in these small competition chambers. And what we see here is that only the fine root biomass depends only occasionally on the competing root biomass. This you see under the interspecific competition in the large figure on the right-hand side under NPK. So it's a significant relationship. And this is what we would have actually, or I would have expected for a long time, that if you have a competing system, roots competing for the same soil spot, the same resources, that they influence each other negatively to basically, yeah, up to a maximum, like something like a carrying capacity for my soil spot. But it's only rarely the case. Otherwise, there is no relation. So this makes, of course, or allows the question to be asked, do they recognize each other? Yeah, do they recognize each other in any kind of a way to basically induce a certain plasticity in their foraging behavior into a certain soil spot? More striking is this figure. This is comparing the two competition chamber pairs. Yeah, remember between the two microcosms, there were always two pairs. One was nitrogen fertilized and one was only phosphorus and potassium fertilized. And the left-hand side is a figure for the intraspecific competition. It means, yeah, two species, two individuals of the same species competing. On the right-hand side, you see the interspecific. And strikingly, there is a tight correlation between the biomass distribution in the PK and in the MPK chamber under intraspecific competition. This means in a moment you have a competitor, which is not from the same species, yeah, it's from a different species. Somehow both roots, Yeah, even that they're in independent chambers, have a coordinated growth. And this can, of course, only maybe be explained that it's related to the status of this host plant where the roots are coming from. So the genotype, maybe the carbon status of this plant where the roots are connected to. So does it matter basically if I recognize who is the competitor, yeah, interspecific, intraspecific, and does it matter how much carbon my host plant basically have to determine the outcomes for this space occupation? Yeah, I don't have an answer to this, but these are interesting questions because I think we are only in the beginning to understand how root competition actually works. Yeah, I have a lot of questions, and this is what I mean in this title with the uncertainties. That we need to look at to which extent basically this ontogenetic scaling or maybe individual genotype, yeah, or their fitness in terms of carbon they have can affect the fine root biomasses. So is this an active process? So it's basically there is a mechanism deciding I occupy the soil, I preoccupy the soil maybe, as it's sometimes described in the competition literature, or is this rather a passive allocation response depending on the carbon status of the host plant or the nutrient level available in a pot? Do roots actually recognize themselves? Yeah, there was a lot of discussion about the wood wide web and the connectivity of roots between with symbionts and ectomycorrhizal fungi, for example. Yeah, it's a potential of signal transfer, but we can also think about direct communication, BVUCs, exudates, and so on? Or is it rather an indirect effect of recognizing there is someone else in the rhizosphere near me by indirect resource depletion? And the last question, I talked about it before is already, and I would be happy if someone can discuss it in the chat, in the question and answer later, or direct message me. How heterogeneous are soils in mixtures? Yeah, we tried to find a reference source because my hypothesis would be that soils in this mixed forest would be more heterogeneous in terms of resource availability than monocultures. But I couldn't find. And there's, of course, a question of scale. So what is the amount of scale, yeah, or the micro heterogeneity we need to look at to basically influence root branch placement? Yeah, maybe the system we work in, 1 liter system here, is already relatively big. For those normally not into roots or this kind of analysis, a word of caution: these biomass effects I talked about are very often very age dependent. Yeah, this paper from Xu et al. From China, for example, shows nicely that fine root biomass over yielding in this mixture of Pinus and Cinnamomum is very strong in the 10 years after plantation. Here you see it with PC. Yeah, the mixture is the PC in the middle bar, but basically over time, 24 years, 45 years later, this biomass over yielding effect is declining. So we talked about biomass now, and of course, biomass is a dynamic factor, which also asks or allows us to ask, do we sample at the right time? But it was very nice that Shiwen Guo last year in the B-Tree published an article where she looked at the different root productivity and the root mortality in the B-Tree plot. Yeah, on the left-hand side, you see again the biomasses, and she looked at different productivity. And what we see is mixing affects at least partially this root dynamics on an annual base. Yeah, for example, the observed root production in root length, here millimeters per square centimeter root window and year, in the Quercus carpinus mixture, for example, topsoil was significantly lower than what you found in the monocultures. Yeah, while the APTC showed only a trend in the opposite trend, and there was basically virtually no significant difference for the 4-species mixture, the oil plot. The same, similar patterns, but not significant, occurred for the root mortality. If you have this now dissolved a little bit into the yearly pattern, you see that there are, again, left-hand side, the monocultures, on the right-hand side, this times the mixtures, different patterns when root growth, when root grow. Yeah, all the different curves on the left-hand side basically show you the root production dynamics over 1.5 years, she could do the study. And you see there's quite dynamic in the beginning of the growing season. Here's day 50, day 80. So we could talk about March, something like this. There's a lot of activity happened already. So there were peaks of root productivity. Later in the early summer, this occurs again. And if you look on the right-hand side in the mixtures, basically, we have this early spring, late winter peaks. We have them at the reduced, but a similar peaks basically in the early to midsummer, basically in the mixtures, and also in the second year. So basically, this mixture dynamics for root growth or for the productivity will basically a function of the individual species. Not so much for mortality, which is shown in the bottom, where you see very distinct peaks for certain species where mortality occurs. Mortality means root disappeared from the mini rhizotons, turned black, basically are assumed to be dead. So basically, our biomass is the function of productivity and mortality. And on the right-hand side in the bottom, you see basically the function for the root mortality across the mixtures, where it's very a nice homogeneous line. So the mortality pattern in the mixtures is much more equally distributed over the different measurement periods than in the monocultures. So somehow there is a smoothing effect, which of course can also be again distinguished or related to maybe the homogenization effect of different plant species. And this, of course, allows us to ask, when do we need to sample? Because we see a lot of fine roots are born and so on, and certain times, and they will totally change the trait pattern of our fine roots. So I want now to come to the second topic. So the root trait variability and plasticity, which is very important because, of course, it covers a lot of species interaction. In the classical niche theory, which is we have a subset of n-dimensional environments, and we say that there are gradients in which a species can survive with interactions. You see on the right-hand side, for example, how could it look? Different monocultures with relatively similar trait profiles, and if we assume that species can only survive in combination, we would assume that in the mixture, this trait profile somehow diversify. Diversify both in means and maybe the variability of the specific trait. Because this intraspecific trait variability has, of course, a lot of effects on ecosystem level responses in mixtures. Think about water infiltration rates, where it's of course not only important that I have a mean diameter, but it matters that I have a combination of large and smaller diameter pattern. And we set up a theoretical framework where we said, okay, maybe we can hypothesize there are four different reactions a plant can have in these traits in relation to competitors. So either the trait can show no competition, no plasticity effect. It can only vary in the internal trait variability. So basically the mean is not shifting, but the breadth, the width of this trait, we can have a shift in mean, and we can have a shift in mean and internal trait variability. First, we checked if there are significances in the overall trait or internal trait variability in total, and you see there's not an awful lot of differences in the variability between the species. So all of them have a certain type of variability in their traits. Maybe here Aether stands a little bit out for root nitrogen content, specific root area, or specific root length. But in general, there's nothing you say one of these four species already an awful lot more variable than the other one. So did the mixing increase this trait dissimilarity? And again, there's a very nuanced answer. It's very trait specific. So what we very often find, and it's following basically textbook advice, is on the top you see the specific leaf area. And for specific leaf area, as a classic example, you see in the monocultures, the leaf area, specific leaf area is very similar between the four species. The moment they grow together in a mixture, you see that it's diversifying basically. Some of them get basically higher and some of them get a lower specific area. For the specific root area, we find only a tendency. It's a little bit similar because Carpinus here you see has a significant broader range of specific root area values, but otherwise they're still relatively similar. For a third trait, like the ectomycorrhizal morphotype richness, in general, they're not more different. They're more similar. And this reflects, for example, also the data I showed earlier today, where the ectomycorrhizal communities in the mixture became actually more similar in the diverse forest. Yeah. So is there a general pattern of this trait adjustment in the community? Or maybe it's related to somehow the society, the status of the plant, related to the competitiveness, the grossness. Here we can see that the relative trait differences basically are different between above ground, below ground, and all traits. Above this dashed line, it becomes basically a trait becomes more dissimilar in mixture, and below the dashed line, more convergent in the mixture. And you see that this differs if we look at communities of 4 species. Including the Acer, which is very different because it's an acquisitive species. If they are basically the less competitive species, Tilia and Capinus, plus Quercus, all of them more conservative species, or really only the losers of our competition, basically here, Capinus and Tilia inside. So the inferior competitors, they possess basically the most convergent traits. This could, for example, indicate that they occupy basically niches, similar niches, which are maybe left by the superior competitors. So if we look at a large spectrum of traits, for example, we can, above ground and below ground, that we see again, the competitive superiority, we think, resides in less trait similarity. And the competitive inferiority resides rather in increased trait dissimilarity. Interesting, like a species like Quercus is very known in Central Europe to have a very strong above ground competitive ability and be a rather weak competitor below ground. Many studies compared oak in competition with Fagus, with beech in the system. And this is actually now reflected here also. There is a significant lower trait dissimilarity or plasticity in this dissimilarity above ground, while below ground, Quercus is much more plastic and adapting this. Coming back to these reaction norms, we found indeed these reaction norms in different traits, so very trait specific. For example, root tissue density would be a neutral trait, it's not changing at all. We have the branching density, so how many side branches I have is rather basically changing in the variability. Changes in the mean, for example, here very strongly in oak realized, and specific leaf area, and classical example for changes both in mean and in the variability of my trait in Tilia. And of course, we were unsure if this trait basically are realized also, if it's an universal thing or it's really only from B tree. This was so nice. I looked now to compare this a little bit with the mesocosm experiment, for example, here for the specific root area. And you see that at least we don't have the same species, but we have Acer in both of them. And you see that in the Acer specific root area of Acer is similar unresponsive as actually in the B tree experiment. Between isolation, intraspecific and interspecific competition, there's not a lot of plasticity going on in terms of specific root area. So Acer obviously don't modify these morphological traits. While for more conservative species like Fagus, in many cases, in many instances, very similar also to Quercus or Capinus, basically we see that there are both changes in means and in the variability with the competing neighbors. Basically the variability is reduced, and also here in specific root area, also the mean basically is lower in Fagus under interspecific competition with Acer compared to isolation or the mixture. Similar happens for fine root diameters, basically, where we see a significant, at least under higher nutrient conditions, as it also occur in the B tree experiment, a reduction in the variability. And this is the same what we found basically in the B tree experiment. So there seem to be universal reactions, at least of certain species, how they react to competitors, because two independent experiments. Of course, all these traits we studied, they're very different in their plasticity. This is the PhD, so the phenotypic dissimilarity index. We could not apply in the B2 experiments because we have, not species individual specific data, but I only use it to show that all these different enzymes, for example, morphological traits, root respiration, they have all kind of different plasticities. In particular, you see very often here, for example, is the specific root respiration, higher plasticity in Fagus compared to Acer. We try to make a general hit list, basically, if physiological traits like exudation of specific enzymes are in general more plastic than morphological traits, but we could not find a convincing evidence. But what is nice and what we would have expected also that two of the enzymes, so NAC and LAP, both of them related to nitrogen mining, basically, they are highly plastic in our systems. Where are they? Here's the NAC and here's the LAP, and this would be expected because we had this divergence between the N-fertilized and the non-N-fertilized. A little bit surprisingly, between Acer and Fagus, the same traits have a bit the same plasticity. So obviously there is something phylogenetic underlying that some functional underlying that some traits can be more plastic than other traits. Among all the traits, specific root respiration is one of my favorites because it's a very integrative trait. We see that there are high influences of interspecific, intraspecific competition, both in the means and the variability in both species, actually. And it's such a great trait because it's so dependent on different subtraits, basically. It's a composite trait, the root nitrogen content, the tissue density, the diameter, specific root area, all of them somehow play a role in determining this. But again, we see that specific root, such an integrated trait, is highly dependent on this competitive interactions, while what we had before on the nitrogen, on the biomass distribution, for example, here the different fertilization regimes didn't lead to a significant different root respiration rate. So really the interaction of different competitive neighborhoods, times, resource acquisition made the trick. So many of these traits are correlated between, and Douglas, give me another 3-4 minutes, then I finish, correlated. So between competing roots in the same competition chamber, the beauty of this. So then we need to ask, for example, here is the average fine root diameter. So the average fine root diameter of really unrelated roots, it's one root coming from plant 1 and one root coming from plant 2, and they are related in their fine root diameters, and not only under intraspecific condition, but also under intraspecific competition. All of these factors are significant, which means, or which explains for me, there is some kind of function of the resource availability, basically, which coining this trait rather than some plastic adaptation to the specific neighborhood. So what are we measuring? Are we measuring actually competition, or are we measuring a function of competition on the resource availability in a certain soil spot? A little bit different again for this nice trait root respiration, where there were basically less clear relationships for the intraspecific cases, but under NPK, under well-nutrient supply conditions, very well correlation for the interspecific condition. So really, if the specific root respiration in Acer was higher, it was also higher in Fagus. So there must be somehow a relationship between the nutrient ability or resource availability in these chambers and the identity of the neighboring competitors. Again, you saw there's a lot of uncertainty remaining. So we don't know or at least I don't know, maybe you can enlighten me in the discussion later. What are the drivers of these phenological changes really? We thought it's a root interaction, but is it passive? Is it active again? Is it dependent, of course? And this is what we saw. It's largely dependent on the trait, but are these drivers actually depending on the traits? Or can we predict, or would we in the future be able to predict these changes, this plasticity, based on different life history strategies, being acquisitive, being conservative, on the competitive status, somehow related to the carbon supply maybe of individual genotypes? what is actually the scale of resource heterogeneity, and there I also would be very happy to receive feedback from the audience, which we need to study to look at scaling effects on different entities. Yeah, roots as a very specific compound organ, yeah, of root tips, second order, third order, and so roots. Yeah, and how these competitive interactions or gradients of resources change the trait interrelations. Yeah, this is something we could not look at. And what are the roles of mycorrhizal symbionts? Yeah, partially taking over the functionality of roots in many species in this game. Yeah, how in the end, of course, the question how all this influences the resource uptake or use efficiencies of my species, allowing potentially a stable coexistence. Yeah, thank you very much for attention. I think you saw that root ecology remains a very exciting, very fresh field where a lot of research can be done by younger researchers in the next decades. But I would really encourage you to consider roots. Yeah, I strongly think that roots and their plasticity, their function, they don't receive the attention yet in the international community they deserve. Yeah, thank you very much, and thank you for all the collaborators in the past years, which allowed, of course, this research to be conducted.
Great. Thank you very much, Boris. That's a great start to the year. So we have now some time for questions. What we normally do is write the questions into the chat box. If anybody really wants to say the question themselves, they're welcome. Just keep it precise. Does anybody want to start? If not, I will start. Okay, wait. Sorry. Yes, there is. Can you see this, Boris?
Yeah, ASAT seems to be more rigid in terms of adaptations depending on mixtures of fertilization compared to Fagus. Still, root respiration was responsive to treatments. Does this mean that only selected traits of Acer are not plastic, but the species compensates it by higher plasticity in other traits? It's a wonderful question because Acer is a puzzle to me, because we really saw in terms of biomass allocation, Acer is not giving a damn on who's the neighbor. It's just growing and maybe and not be very plastic. So why should the root respiration be plastic? Maybe in the end, it depends on some kind of a carbon limitation, because of course there is the carbon expenditure for all the structural things and for basically the maintenance procedure. We know that particularly fine root systems are notoriously expensive in terms of carbon. So I could think that basically Acer also will need to save at a certain place some carbon, and maybe if it cannot do this in the structural carbon, basically, in adjusting his allocation of carbon between roots, shoots, and so on, relative growing, as long as there are nutrients, maybe it has still the ability to fine-tune this basically by the activity of exudates, everything, or uptake processes, maintenance processes, everything which causes basically root respiration. But it's only my hypothesis, basically.
Okay, other questions? Then I will ask one again. Maybe I can get it in this time. Going back to this Acer, it's the only one that has a relationship between above ground and below ground biomass, or fine root biomass. But it's the only one, if I'm correct, that's diffuse porous. Is that the reason? Is it because when we look at above ground biomass in ring porous species, there's a lot of dead tissues, right? A large amount of the biomass is not being used anymore, whereas in Acer, it is being used, I think.
Yeah, I mean, I don't know if this is universally true. Actually, I don't remember that I saw somehow these allometric equations for older Acer species. This depends basically. So this would mean that at a certain time when Acer starts also to have less conductive tissue, basically, it would also reduce the amounts of roots relative to the standing biomass. So only in the beginning now, in these first 10 years, where basically all the sapwood is active, it means it needs more roots to supply, yeah, or to be able to supply the water to fill everything. Yeah, might well be that is one of the reasons, or just a lack of adaptation, because it's so, we know Acer drives very much on this very rich soils, and of course there are some consequences of this large carbon expenditure. It's not very resistant to fungal pathogens, for example, and all of the Acer stands die. So there is a trade-off, of course, of this growth maximizing strategy. Yeah, if some people call Acer the new spruce, basically, or what foresters hope for. And other important traits to secure your survival. Yeah, it's maybe no surprise that there are no Acer monocultures naturally. Yeah,
yeah, could be. There's another question. I assume it's Michael Scherer-Lorenzen. Can you see it, Boris?
Yeah.
Okay.
So Michael Scherer-Lorenzen from Freiburg wrote, your question on soil spatial heterogeneity and mixtures. He asked, we usually see species-specific kernels on litter fall around single trees with clear zones of litter overlap between neighboring trees. Thus, I guess this spatial scale should matter for nutrient cycling and root nutrient uptake. This is a good question, the spatial scale I'm referencing to. I always think, what is the unit of soil? I boldly claim there is an overlap of root systems. But of course, in the bee tree, for example, this overlap of root systems, this was taken from a sample with a 7 centimeter core to 20 centimeter depth. Yeah, before, when I did this in Germany before, maybe we did it in 10 centimeter increments in a 5 centimeter core, but it's still, some 100 milliliters of soil volume. So what is really the micro scale? Yeah, let's say on the distance of centimeters, we should actually look at specific acclimation effects of roots to hotspots of soil nutrients. Yeah, I mean the fox peat somewhere, for example. Yeah, or what you're referring to, I think, if it's really, if you talk about the canopy size, I think it's a larger effect than I mean, because we see already here in this microcosm effect, the spatial effects of neighboring microcosms from the same plant. We talk about distances of 10 centimeters basically. Yeah, and I assume that similar heterogeneity is present also in forest stands, and that it's maybe amplified in mixed forest. Yeah, of course, somehow stratified. This is a very good point by where the litter occurs. But think about below ground litter. Yeah, we don't have a very good understanding how far these root systems actually extend. Yeah, so the root litter and the associated mycorrhiza of a specific species. What is the extent of this remains relatively unknown. Yeah, only in the beginning, 20 years ago, we did this. So this was far beyond the canopy, of course. So this classical knowledge, the root system ends approximately where the canopy ends, of course, is, as you know, doesn't hold true for roots. Yeah. So it's a good question. Yeah, but I'm struggling. And if you had any data on microscale changes, pH and whatever, I would be very happy to see it.
Okay, Boris, there's two more questions at the moment. One, it must be Hoike Kreiger. Again, can you see this?
Yeah.
Yes.
So Hoike Kreiger, professor, wrote, what was the definition of fine roots in different species? Since they differ greatly regarding morphology, structure, and sizes, and also since ectomycorrhizal species take over a number of functions, it seems rather logical that the endomycorrhizal species, also Acer in this case, produces more fine roots. Has the ectomycorrhizal been considered in correlation? Yeah, we were hoping that, of course, we had a different hypothesis basically when we planted this bee tree experiment, for example. We wanted to create with the two species mixture actually a rather endomycorrhiza and a rather ectomycorrhizal dominated patches, which failed a little bit because we know that Tilia or at least we thought we know that tilia could be colonized by both endo and ectomycorrhiza. But this turned out to be rather ectomycorrhiza. So this faded a little bit. And also, as you saw, that the ectomycorrhizal diversity was reduced. No, we did not relate it really to this. Yeah. When you ask about the definition of fine roots, it follows the classical Böhm classification I grew up with from Göttingen, which is relatively okay for us because the here studied species, they are relatively close to the optimal it was defined on. So it is this threshold of 2 millimeters for fine roots really applies, to the best of my knowledge or what I observed over years in the field, to the absorptive function. Yeah, so we were lucky basically. If we had a species like Salix or something like this inside, of course, we could not choose a simple diameter-based cutoff point for this transition between absorptive fine roots and transport fine roots. So I think the fine roots we studied here are all absorptive fine roots. Overwhelmingly, yeah. For the Fagus, it might sometimes they are a little bit thinner than 2 millimeters.
Okay, there's one more question. Which is from Rich Norby. Can you see it, Boris?
Yeah.
Okay.
So Richard Norby wrote, the very high diversity of tropical forest makes it virtually impossible to analyze species-specific responses of fine roots. Hence, almost all analyses are at the root community scale. When changes in some root rates, such as specific root length, are observed in response to a manipulation, it's difficult or impossible to determine whether this reflects plasticity within a species or a change in the species composition of the community. Yeah, I see it. I see a solution to this dilemma, but unfortunately, my gotcha proposal in the Czech Republic was rejected end of December, where we proposed to develop actually a methodology to have a machine learning approach to classify them relatively easily. And I think this is really something. So if you can get your hold of a little bit of money, machine learning image recognition is so advanced now, and it's working so well that I think what we're missing is basically the training data set of different root periderms. Yeah, this is if we classify them basically in the laboratory, it's still one of us sitting there with a stereo microscope and classify them. And in the European system with maybe maximum 11 species, it works relatively well after. Yeah, we totally understand that in tropical systems does not work. But I would really count, I'm very technology positive in this case, that we could, as a community, develop a training data set for different species and solve this problem by automatically recognizing them. When even think about robotics, it must be possible. Yeah, and this is for sure the root science 4.0, that there is in robots sorting our root samples. It's not rocket science. It's only a question of a little bit effort and maybe a little bit more money.
That's an excellent place to end. There are two more comments. One is from Clara Marek, who's showing us a new paper, so everybody can click onto that. And one is a comment from Hoika again, showing that she did something like this over 30 years ago. It's always worth remembering for many people out there that actually a lot of this stuff comes around again. So, but thank you very much, Boris. Excellent start to
my pleasure. Thank you very much
to the year. Just to finish off, just a few things. The next seminar is on the 11th of February, which will be Lars Vesterdahl, and he's talking about carbon sequestration. Thank you very much again, Boris. Thank you for everybody for coming. We had over 70 people. Not bad start for the year. See you on the 11th of February. Bye-bye.